And thanks for coming. Thanks for those of you that are around the building, and I hope that more people find the building in the actual fifth floor soon. I'm Julie Barnes-Wise, for those of you who don't know me, and I'll talk briefly in a minute about the background of the Global Healthcare Innovation Alliance's project. But this symposium is an offshoot of that, and we've been working basically trying to track all the different alliances, first around the Ebola alliances, and now, of course, moving into Zika and all the different things that those entailed and the different implications for about a year and a half now. I think there might be one student here from last year, so this is the second year's worth of students that have actually worked with me on this, and several of my younger colleagues, Anna Santos-Richmond, who is hopefully watching this on streaming, couldn't be here. She's out of the country, but she's going to be watching and participating as we move forward. So, if anybody's tweeting this, it is #GHIA. We have a website that supposedly launched about midnight last night, and that's on there. And if you want copies of presentations, please sign in. I'm not sure it's a pad of paper out there to sign in on, but we need to put one out there. And thank you to our sponsors. If we didn't have the sponsors, there wouldn't be food for you all to eat this breakfast. We probably wouldn't have a lot of setup, and it would be a much different symposium. So we appreciate Duke Science and Society, Trent Center for Bioethics, Humanities, and History of Medicine, Bavaria Nordic, and Marshall Christine and Forum. Thank you. I have to thank the team. These are students who have worked constantly now for, as I said, a year for some of them and over a year for some of them, and the project would still be a gleam in my eye if not for all these students. So thanks to all of them. There are several posters. I think there's 2 posters up now, and another one is going to go up a little bit later. So their individual work has informed some of this project. I told Dr. Rick Ballou last night that it was nice to finally meet him. I think I've been stalking him now for about a year and a half, and I've talked to him on the phone a lot of times. But about the time that the Ebola outbreak started to become publicized, I probably had a strange reaction, but my reaction was I was fascinated by, at first, the GSK Alliance consortia that started to be formed around it, because my background is in drafting, negotiating agreements, and the business development part of forming partnerships. And I know that it takes a long time to do one agreement. One license agreement often takes a year to get done. His business development person at GSK Vaccines told me, we were on a panel in New York last October, they had 50 agreements, 25 of which were key. And I think most of them got done in about 4 or 5 months, and that was extraordinary. So that's what first caught my eye. And of course, as you'll hear today, there's many other aspects of those alliances and related aspects, including incentives, that are very interesting. But that's where we started, trying to find out kind of how did it happen, and who was involved, and what were the problems in forming them, and what went right, so that hopefully for all of the next infectious disease outbreaks, including one that came much sooner than any of us expected, we'll be able to accelerate the process of forming alliances to move medicines and vaccines forward. So this is just, obviously, we try to figure out who all the major players were and look at the critical issues facing these alliances, and develop kind of questions for them. And so we were tracking people kind of around the globe, trying to get them to tell us almost in real time why they were working on developing Ebola vaccines and what was the process of forming these alliances. What we've transitioned into a great extent is development of partnering tools and examining incentives around this. So for the future, for those of us that do actually work to draft and form these agreements, that we hope that we and those that hopefully we can share this with can move through the process much faster. So obviously, you all think the Ebola outbreak was somewhat important, or you wouldn't have found this room. But from my perspective, first thing, as I just mentioned, is I thought the Ebola outbreak resulted in new and broader alliance creation than had really been seen before, and certainly renewed appreciation for the need for system improvements, because everybody that participated in any aspect of this, and I know Rick's going to talk about this a lot, was frustrated by a lot of the inefficiencies of things that slowed down the process, and in some cases slowed down the access to data. And then the fear factor from an outbreak, for better or worse, that was a big driver of financing and within companies getting a lot of things done faster as politicians put pressure on companies and companies put pressure on their own employees to do things. And also, from my perspective, there's also a study access to medicines issues, and it seemed to me that the reaction in the US and Europe to the fact there was not an Ebola vaccine, and now to the fact there's not a Zika vaccine, brings home a little bit to us what most of the rest of the world faces, which is they don't have access to vaccines and medicines for diseases that affect them. And we're not used to that. We're used to, we get sick, we go to the doctor, there's a vaccine or medicine. And so I think Ebola was sort of the first thing where people were horrified that there was not something to treat them. And that is, as I said, what a lot of the rest of the world lives with all the time. And I do have to give a shout out to GSK, who made an announcement yesterday that they are going to be, in some cases, not enforcing their patents, and in some cases not filing patents in lesser developed countries, and also sharing their oncology patents with medicines patent boards. So that's great news. Finally, and re-energizing the participants who are fighting infectious disease outbreaks. These people, and including many of you, have worked so incredibly tirelessly for the last couple of years on this. And for those who haven't been involved with this, talk to them. Most of them haven't had vacations, and they've been working really late hours, and they've dragged teams from lots of other projects to do this. So I'm humbled by all the things that they've done. So as I said, now we're focusing on development of different provisions and model agreements, and trying to increase the way that the process of forming alliances happens, and to examine and develop incentive mechanisms across the different stakeholder types. And David Ridley's going to talk to you a little bit later about their incentive system development and what that might mean for the future and what additional incentives might develop. So these are my goals anyway: to share across boundaries, to learn from those who have built Ebola alliances, discuss the outstanding issues, determining the next steps in fighting the next infectious disease outbreaks, and each of us from our own perspectives, and have a little collaborative learning. So this is what we hope for you. That you're going to enjoy the speakers and provide your own perspectives and ask questions, and help us develop what we ought to be working on next to help the process move forward more quickly. So, finally to the speakers, and I'll stop talking. I must say first, Adrian Thomas apologizes for not being here. He actually lives in Brussels, and he's stuck in Brussels, and so we're all thinking of them because they still can't, the airport is even still closed, and most of them are living with a lot of fear right now. So, all right, so I'm going to introduce our first speaker, Professor Artie Rye, who's the Elvin R. Laddie Professor of Law at Duke Law School and the co-director of the Duke Law Center for Innovation Policy. She's an internationally recognized expert in intellectual property law, administrative law, and health policy. She's also taught at Harvard, Yale, and the University of Pennsylvania Law Schools, and her research on IP law and policy and biotechnology, pharmaceuticals, and software has been funded by the NIH, the Kauffman Foundation, the Wilson Center, and she's published probably even more than this now, over 50 articles, essays, and book chapters on IP law, administrative law, and health policy. I think I'm going to stop there. And just say that she also regularly testifies. She regularly testifies before Congress, and I'll let her give you her educational background if she chooses. But I'll stop. But welcome.
Please.
Do I need to?
No, I don't have any slides, so I was going to come up there and talk. Is this being webcast?
Yes, it is.
Okay, great. So I will speak into the microphone.
Speak into the microphone. Yes. That's a word for you.
I already drank from. Very assertive. Great, so thank you so much, Julie, for the more than generous introduction. And For those of you who are coming from out of town, a warm welcome to Julie. It is warm, but I'm afraid it's not as sunny as it usually is, so apologies for that. So I know Julie and her GHIA team have been working really hard on developing a legal infrastructure for alliance formation, particularly in the case of emergency infectious disease outbreaks. And I know that because several of my law students have been working very hard with Julie. I won't embarrass them by naming them right now, but I will say that we're also recruiting law students today. Today is our admitted students day where we try to promote Duke as a great place to be. And one of the things I will talk about, again without naming you guys, is the way that students can get involved in really important projects that matter in terms of saving lives. So you guys will be invoked again, for better or for worse. So I have to confess that I'm not steeped in the complexities of global infectious disease issues on a day-to-day level in the way that Julie and her team are, and our other speakers today clearly are. So I'll try to keep my remarks relatively brief, and I hope they won't be too uninformed, but apologies in case they are. What I have thought about and written about a lot over the years, as Julie's introduction perhaps indicates, is barriers to collaboration generally in the biopharma R&D space. And I'm sure that it's no, I'm not telling you anything new when I say that barriers to R&D in this space are probably uniquely difficult. I've also looked at R&D collaborations in other spaces like software, and there it seems that for a variety of reasons that I'll talk about in a minute, you can get collaboration that's sometimes a little more organic, less heavily lawyered, and so forth. And in this case, the collaboration tends to be heavily lawyered, probably for good reason, but you know, that's the reality of the situation. And so I'm really excited to see the enormous effort that Julie and, again, my shout out to my students and others are putting into addressing at least two key interrelated issues that are, unfortunately, for better or for worse, often lawyer heavy, let's put it that way. First, of course, contracting, and second, less lawyer heavy specifically, but related, incentives and funding. And I think these issues have to be lawyer heavy for better or for worse. So I'm really excited that Julie, who has so much background in negotiating contracts in the real world, is coming and helping us academics who kind of think about these issues in a much more theoretical fashion, really work on the practical stuff, because I think the practical stuff at the end of the day is what matters. Much as I'm an academic and I like to think about theory and empirics and large-scale data sets and all that, practically speaking, what Julie's doing matters so much more. So just a quick word on theory, because I can't resist. So contract theory would tell us that the costs of collaboration in this industry would be extremely high in terms of the lawyer costs, essentially. If there are high capital costs involved, and whenever you have lots of money involved, you have lawyers. And then, of course, the heavily regulated nature of the industry. It's very heavily regulated, much more so than many of the other industries where we see R&D collaborations. And that's probably a good thing on balance, but the nature of it means that you're going to get many more lawyers involved. And there have been large-scale empirical studies, not as many as we'd like probably, but at least a fair number that have shown that the R&D alliance contracts, to the extent we can get access to these contracts as scholars, they're much more complex in biopharma than they are in any other industry. So they're very complex governance and dispute resolution structures. I'm sure this is all not news to any of you, and very deliberate, careful allocation of intellectual property rights, data sharing, access questions are very carefully negotiated as well. So, and as I understand it, the prior work by Julie and GHIA shows that this actually happened in the Ebola context as well, that there had to be a lot of negotiation around intellectual property, data access, liability, and risk, including for clinical trials, management and control of the alliance. So this is unfortunately even the case where we have emergencies that are facing us. But of course, the reality or the need for compressing the time window in these cases is quite high. So it looks like in the Ebola context, at least some of that urgency was felt by the actors, and that's good. One of the challenges I presume in this space is that it may be a little more challenging given the diversity of the actors. In a lot of biopharma alliances, we're just talking about the private sector, whereas here we have a major role for government agencies and NGOs. So as a consequence, as I'll mention in a moment, incentives are likely to be quite diverse as well, and contract theorists will tell you when incentives are diverse, negotiations are even more difficult. So all of this is to say that I really applaud what Julie and GHIA have been doing in terms of gathering up as much information about agreements as possible in the hopes that we can create templates that can serve as starting points for future negotiations so as to get these done quickly, as quickly as possible. I can tell you that in one specific context where I've had some real world experience in a particular NIH-funded public-private partnership to rescue failed drug candidates, essentially take them out of the small molecule libraries that pharmaceutical companies have and expose them in interesting ways to researchers in academia. The template contracts were really critical. Without the template contracts, nothing would have gone forward. Basically, everyone would have just figured themselves to death, and that would have been that. So I think Julie is getting exactly the right set of issues, and I'm so excited about that. Just a quick word on incentives. Again, you'll probably know more about that than I in this particular space. But good contracting, in theory at least, is supposed to align all the incentives properly so everyone is incentivized to do the right thing and not shirk and not be opportunistic and all that stuff. And presumably in this area, people are also motivated by humanitarian objectives that might lead them not to shirk and be opportunistic and generally just be bad actors. Let's hope so. It's also, I think, very exciting in this space that we do have a prominent role for NGOs and funders who can use, as I was Dick Wilder earlier, the power of the purse to promote good action. And that, I think, even more so than government funders who are restricted by all sorts of regulatory and political barriers, and believe me, having worked in government, there are many of them, to really using the power of the purse. NGOs can use the power of the purse in ways that I think are much more creative and intelligent, frankly. So all in all, I think that highly practical efforts like what Julie is doing are incredibly promising, and I'm personally really looking forward to hearing from the speakers today. I've already heard in the past a little bit from folks like Dick Wilder and David Ridley, who came up with the priority review voucher idea, but there are many more folks I haven't heard from, and so I'm really looking forward to it. So again, welcome to Duke, and at least it's warm. Thank you.
Thank you for the kind words. Our next speaker is Ross McKinney, who is a professor and practicing pediatric oncologist. Oh, got that wrong. Infectious diseases. Okay. He's director of the Trent Center for Bioethics, Humanities, and the History of Medicine, and he's going to bring the ethical element to infectious disease outbreaks. Switch to the presentation. All right. I was trying to get the presentation, but I think that it was fine. Okay. Thank you.
Well, thank you very much, Julie. I'm delighted to be here. This particular talk came out of a project, which was a course that I taught in the fall. The course was on ethics in epidemics. And I thought that was a really interesting opportunity to evaluate a fundamental issue in ethics, which is the balance between individual liberty and how societies address the balance between individual liberty and the collective good. And that epidemics give an opportunity to understand that because the pressures are placed that are high. And you begin to see how the society is going to respond to that balance. And my hope was, by looking at a series of epidemics over time, beginning with plague and ending with Ebola, that we would see the evolution of society. And what I learned is human beings are human beings. And we are no better at addressing the issues of individual liberty and collective good at the present day than we were when we were dealing with the plague. And in fact, probably the single best group was the city of Eve in England in the 1500s, which addressed the plague by isolating itself. It's probably a more noble gesture than anybody has done since. In any case, given that basis, I got really interested in this question of ethics and epidemics. So what I'll talk about are some of the ethical dilemmas that are posed by epidemic diseases, a little bit about the basics of how we make ethical decisions so that we have a framework. I'll talk about the power of urgency, because what epidemics bring that drive a lot of the decision-making is a sense of urgency and need, and we'll also talk about what I consider to be the equally balancing power of uncertainty, and so that we make our ethical decisions in the context of urgent situations where we don't know what the hell we're doing. And those two pressures create drives that often lead to bad decisions. So what is ethical? People often say, give me an evaluation. What is this ethical? And the reality is, what is ethical is very much situationally dependent. There are not good rules and structures to make the decision. There are different perspectives, different ways you can look at ethics, and different frameworks we can use. And which framework we are using starts to determine how we respond to whether something is ethical or not. And then you layer on laws, which are codification. And I'll say, as people have often heard me say, what is legal is not necessarily ethical. What is ethical is not necessarily legal. The laws are a code that is an attempt to create a framework that can be used repetitively in multiple situations, but often they are not applicable to the specific situation that's being addressed. So, same thing. Laws are created to codify values and make the application of those values consistent, but it's a blunt instrument. And ethics and regulations are often not congruent. And you have to remember that. And the question then becomes, do we not do what is regulatorily required? Do we follow the regulations and laws? Or, even though it seems wrong, or do we try and change the regulations and laws, which is often difficult because we build such difficult processes around the change of law and regulations. And when there aren't laws and policies, what do we do? How do we decide what is right and what basis do we use? So one of the core bases that typically is used is the deontological principles. So a deon is a duty or obligation. Ology is study of. Deontology was Kant's worldview that ethical positions that judge the morality depend on how well an act complies with ethical rules and expectations. The act is the focus. What you do is the focus. And in general, in deontology, the rules tend to be absolute. There's a little waffling sometimes, but really, rules are absolute. And this is where religious fundamentalism comes from. So if you want to see the full application of deontology, look at fundamental religious groups, because they are trying to achieve that sort of rigid application of rules and laws. What is right, as I say, is defined by the act, and there's Kant's categorical imperative, which gave a good framework for trying to decide what is right. In a situation, act according to rules in such a way that you wish that your action would be transformed into a universal standard. So when you are asked to make a decision that relates to an ethical position, make your decision in such a way that it would be consistent with what you would hope would be a universal standard in the future, as a way of trying to define what is the right thing to do. And another key element is going to come back in our discussion about epidemics. Do not treat people as a means to an end because they are the end unto themselves. So don't ask people to be part of a research experiment, for example, without agreeing to participate, because otherwise you are using them as a means to an end. And the way we get around that notion of using people as a means to an end is to ask, are you willing to participate yourself? Then they cease to be a means to an end. Instead, are participants in the research process, which is why the language we use now about human subjects is often about participants and volunteers, because we are trying to get their agreement to be part of our means to an end. The focus on, again, the nature of the act rather than character, and virtue will be focused on character. So we're going to have there 3 stages. There's the act, there's the consequence of the act, and there's the character of the person who is engaging in the act. And those are the 3 ways you can look at how ethical decisions are made. Laws are much better at dealing with the act than they are with either the consequences or the character of the person. They tend to focus on deontologic principles, tend to focus on things, and laws tend to focus on the act. It is forbidden, it is allowed. Consequentialism. What is right is defined by the consequences of the act. So not the act itself, but the consequences or the intended consequences of the act. And the one that most people know is utilitarianism, which is greatest good for greatest number as the goal of an ethical. So a well-made ethical decision is one that does the greatest good for the greatest number of people. And we know the exemplars of consequentialism, Jeremy Bentham, John Stuart Mill. But in its pure form, it really doesn't work very well, because people will exploit consequentialism. And the classic case you can look at is, the transplant patient. You have 5 people in need of a transplant. You have one person standing right there who happens to match all 5 of them. They all have lethal diseases. One needs a heart transplant, one needs a lung transplant, one needs a liver, 2 need kidneys. And you have one person here who is a donor to all of them. Greatest good for greatest number says you sacrifice that one to save those. We don't do that because we don't allow consequentialism in its pure form to drive us. We always leaven our consequentialist perspectives with deontologic principles. And in fact, what we generally do is constantly balance deontology and consequentialism. And the act of doing that balancing is typically where we think the virtuous person comes in. Because virtue is the person who is able to define and well adjudicate between the deontologic and consequentialist perspectives. So, virtue ethics, Aristotelian, the focus is on the actor. What would a virtuous person, a good person, do in this situation? And you can list virtues. You can make them categorical. But it turns out that virtue ethics isn't very practical. And when you're trying to decide something in a crisis, like in an epidemic, it doesn't work very well. We tend to fall back instead on what I would refer to as a social model of ethics. And the right or good usually comes out of the norms that we have come to adapt to in our lives. They evolve, they have changed, and I think that the notion exists of a moral compass, that there are people who basically know what is right and wrong, and we know that the absence of that creates things like sociopathies, which are definably distinct. So you can see both the presence and absence of a moral compass as a driver of good behavior. And we human beings depend on trust. We depend on people doing what they say they will do. We depend on a certain level of honesty, although we don't expect absolute honesty. The social model, however, is not very useful for defining things. It is what we actually work with and use and every day, sometimes referred to as practical ethics. Is what is the normal way we handle ethical decision making, but in a specific situation, it doesn't give you guidance, it doesn't give you rules. And in fact, what I think we find is that although you can define these various strategies for defining what an ethical life is and what is ethical, the reality is that when we get into a situation, when we get into a context, that contextualization turns out to matter more than any one set of rules. and then we do our best to navigate balancing. So the original question, what is ethical, to my mind, turns out to be fraught with problems. We simply don't have good tools to define what is ethical and what is not ethical. And I know that sounds uncomfortable, but it's my perception of what the reality is. So we confront these crises with difficulty defining what really is ethical. So I'm going to give you an example. You have a prototype Zika virus vaccine. It uniformly produces neutralizing antibodies after 2 doses in healthy, non-pregnant adults. And a recent study that most of the people who have probably read in New England Journal suggests that 29% of Zika-infected pregnant women have significant damage to the fetus. Microcephaly, intrauterine growth retardation, fetal death. So you ask the question, when should we study this vaccine in pregnant women? Now, you can give a series of technical answers, but what's right? You're going to be confronted by the urgency of a crisis, and you're going to be confronted by the notion you do not want to do harm. But at what point do you do harm by failing to act, just as you can do harm by acting? And how do you balance that failure to act against the acting? And we tend to focus, because of our culture, on the act, and to blame somebody who takes an act that ends up with a bad outcome. So what drives a situation like the Zika case that I was just talking about is the sense of urgency. We must do something. We must. And you hear that language of we must do something. A failure to act sounds like a moral fault. We have placed a moral judgment. We are using our ethical language to try and drive something forward. That sense of urgency, we must do something to protect those children. At the same time, there is a sense of uncertainty. Are we going to do harm? Which path should we pursue? Should we do the vaccine now? Should we wait until we have established that it works in the unpregnant woman, so that we know well what its characteristics are before we confront the first pregnant woman and take the opportunity of the fact that this vaccine could, in fact, perhaps even induce, depending on the type of vaccine, negative consequences in the pregnant woman? Are there risks to acting that, in fact, are just as bad as the risks to not acting? So let's go back to that Zika virus vaccine and move to a slightly different situation. Zika is associated, we know, with the serious fetal outcomes, but it's also associated with Guillain-Barré syndrome. And this is going to be probably, scientifically, a relatively easy vaccine to develop. It's kin to other vaccine programs that are already fairly far along. And if you're in the vaccine world, I apologize for my sense of an awful lot of work, but relative to some other vaccines, it's likely to be relatively easier to develop. Maybe we can figure out a killed vaccine approach. We already know how to do killed vaccine. We figured that in the 1950s. The virus itself is already pretty attenuated. Zika, it's interesting, it's a virus that 80% of the time doesn't produce much in the way of symptoms. So it's a pretty attenuated virus, except in the situation of pregnant women and those rare cases of Guillain-Barré syndrome. But when you give a vaccine, you're going to give it to a whole lot of healthy people. That's the basic underlying principle. You're going to give it to a whole lot of healthy people, and they should be better off for having been vaccinated. And rare side effects can turn out to mean a lot in a vaccine. And they can even be very rare side effects. And so you worry what the Zika vaccine will do, either for the pregnant woman and the woman who gets the vaccine and did not know she was pregnant, and it may have some effect on the fetus, or it's an attenuated vaccine, or you might get Guillain-Barré syndrome. And it's entirely possible that even if Guillain-Barré syndrome is 1 in a million, that more people get Guillain-Barré syndrome from the Zika virus vaccine because you give it to everybody than get it from the Zika virus itself, because it's an infrequent infection, at least at present. And even if it becomes epidemic, not everybody will get it. And you say, oh, that's not realistic thinking. Remember that the attenuated poliovirus vaccine was the sole source of paralysis related to poliovirus in the United States for decades. After we had eradicated the poliovirus, the wild poliovirus in the United States, we continued to use the attenuated vaccine. We had somewhere between 5 and 10 cases of poliomyelitis per year in the United States, all attributable to the vaccine. So it is good to use the vaccine to protect people, but there was a price. And what will the price be in the Zika virus vaccine when we have it developed? So the challenge when you're in a maelstrom, like the arrival of the vaccine, when there is the virus, there is a wave of publicity around the epidemic. There is this sense of urgency. We must do something. The government is not acting. Industry is not acting. We must do something. And at the same time, there is this apparent conservatism on the part of the key actors because they're worried. We tend to drive in these situations towards consequentialist thinking. We want to do the right thing. The right thing must be to do some good. So we tend to drive that way. And the companies and the governments tend to fall back on the rules and say, yeah, we need the rules. But people are more willing to overlook rules in crisis situations. So the rules change for companies when you're in the midst of an epidemic. The epidemics arrive acutely. They leap across legal boundaries. That's one of the problems. You look at Ebola, there it was leaping across, Sierra Leone, again, Liberia. There it's leaping across. It's not in one country. The rules of more than one country start to apply. Look at Zika. How many countries is that in now? It is flitting from country to country, and the rules change as you move. So whose roles and whose laws must we comply with and adhere to? What is the liability situation on the research? Does it vary from place to place? Is the company going to choose to do its research someplace where the liability laws are more lax? Will it be willing to do experiments in certain situations? Is it exploited if they do that? Or should they go to the most restrictive environment so nobody can challenge them for having followed all the rules and regulations that were part of dealing with the situation? And what are the ethics if you have something and you have the opportunity to rescue and you don't do it? You are sitting there with a drug which might be a therapeutic against Ebola. Are you obligated to study it? Or should you do more animal studies before you get into the crisis situation? Because that's what the companies were being confronted with in the Ebola outbreak. People were dying of the outbreak. There was a sense we must study it, yet were the drugs sufficiently advantaging to the patients to justify their use in those situations? And in general, since the high fatality rate was so, created such a sense of urgency, the answer was yes, but there had to be a constant rebalancing of when is it appropriate? Are we not exploiting these people? No, we're not exploiting people. We're trying to do rescue, but how do we keep that clear? There is the perception of exploitation by those who look at the pharmaceutical companies as simply profit generators. In fact, their good attempt to do good is sometimes tarred as profit generation. So we see this sense that the ethics of the situation become a little murky because of attribution, because the motivations are not always clear. We would hope that it's a humanitarian impulse that is driving things, but the humanitarian impulse sometimes should be not to try and do things if it's too soon. So if we look at Ebola versus Zika as examples, treatments and vaccines, Ebola was often fatal, created urgency. Treatment for Zika, it's mild, other than pregnancy. We're probably not going to develop a treatment for Zika. It's difficult to develop vaccines or treatments for either disease, particularly for Ebola, because it's so evanescent. The outbreaks come and go. The incentive structure does not favor epidemics which only periodically occur. And scary as they are, the market completely evaporates and was never very large and was never in an area where it had any resources to spend on a product, particularly an expensive product to develop. So you were going to spend an astronomical amount of money, and the only really good reason to do it was humanitarian, because it was not going to be justified by a profit motive. But you have to deal with things like strain variation on vaccines. You're going to have to deal with things like variations in the virus, perhaps the susceptibilities when you develop a drug, all for a disease that's fairly evanescent, although frightening. And then there's the question of our willingness to accept risk in studies of treatment or vaccine. And what level of risk are we willing to accept? And does the nature of the disease change the willingness to accept it? Ebola people are willing to accept. If they perceive themselves at risk of Ebola or have it, they are willing to accept a higher degree of risk in the research. It is adjusted, it is readjusted, and that calibration is a consequentialist calculation. Where are we doing the right amount of good? And if you're going to be in contact with the cases anyway, you probably want to be vaccinated. You'll take your chances with whatever the side effects of the vaccine. You hope that it works, but you want to be protected as best you can, and you're willing to accept a probability of failure in the vaccine, which you might not be willing to accept with Zika, where the risk of the disease is in itself so much lower. We wonder who is at risk for the disease. And one of the things that's interesting in this case is the person at risk, if you do a study of an Ebola vaccine, is the person who is consenting and is going to receive the vaccine. If we are looking at a Zika virus vaccine, ultimately the person who you are protecting is a person who is not yet here. It is the fetus who is the target, largely, of the vaccine program. And so the people who are to benefit from the vaccine program and its development are actually not the recipients of the vaccines. They are benefiting because they're the parent of the child, but there's an indirect nature to it. Are there alternative approaches? Because one of the things when you think about doing a treatment or a vaccine, are there other ways that you could solve the problem? And in fact, if you look at the Ebola outbreak, probably the other way to solve it beyond the vaccine approach, which we've used, was in fact the one that worked, changing funereal practices, changing public health measures. Those were able to bring the outbreak under control. If you look at Zika, what are alternatives? Well, I don't think actually they're very practical. It's going to be attempts to limit the mosquitoes. It's going to be moving indoors if you're pregnant, living indoors entirely, wearing DEET all the time. I will say I did look this up, and DEET is not considered to be a toxin for pregnant women. And for those of you who are not familiar with DEET, you will probably get very familiar with DEET because the CDC revised, at least those of you who are North Carolinians, revised the map, and Aedes aegypti gets up into North Carolina. We were a malaria zone, and it is anticipated that we are potentially going to have Zika getting this far north. And so get familiar with DEET. DEET Woods Off is an easy way to remember. But you will get familiar with that nice chemical aroma that indicates protection. So we should consider, when we think about the kinds of steps that we'll be talking about today, we should consider what the ethical nature of our acts are and their consequences. And I say both the act and the consequences, because really it is the balance of those two things that we should be using to evaluate the ethical nature of our behaviors. We should realize that challenges exist in the epidemic situation. There is that balance between personal liberty and the collective good. There is the constant war between urgency and the compelling drive of the unknown and uncertainty, which means that we tend not to act at times in order to protect ourselves from engaging in an act that we are uncertain as to whether it will in fact create harm. We must balance safety and good practices in order to achieve the most rapid resolutions possible. So I hope I've been provocative, and I hope that the conference is a good one. I'm happy to answer any questions if anybody has any. Yes, Chris.
Comment on what you call the culture of vaccination. So if you look at vaccination as a principle, is it culturally a norm? Is it easy to...
Okay, the culture of vaccination and vaccination as a norm. I think that vaccination has become a in most of the world, people have come to accept there are isolated areas, there are cultures that have rejected it. Northern California, for example. But most of the world is able to understand the price that we have suffered under significant severe diseases and the ability to eliminate them or attenuate them by vaccine and accept that as a reasonable cost. That balancing between the risk, most people don't actually know what the risk is. Most of the risks in the vaccines we use, because we use that standard of we're giving it to healthy people, most of the vaccines we use are very safe, and the side effects are very uncommon. And they must be, because we are engaging in an act in healthy people, and we don't want to make them less healthy people for having vaccinated them.
So one slight change to that is, let's say the vaccine is actually not working.
Okay, malaria vaccine. I mean, it may turn out eventually to be good, but if we were to implement what we have, relatively marginal effectiveness, it becomes harder to justify and becomes a more complicated consequentialistic equation. It probably becomes rational only in the areas where the price is already so high or the urgency is at its highest. And so a marginal vaccine becomes most useful when the urgency level is high and where there is sufficient certainty about the failure to act that you are willing to accept an uncertainty of benefit from the vaccination. Consequences to the equation.
Now, the answer to your question with this was, is this in America, or is this in West Africa, is this in Brazil? Because I will say, in West Africa, they do not want to take a vaccine. There is lots of communication people out there, there's a lot of rumors. You know, you're dealing with tribal folks, you know, that are very undereducated. You know,
But I think
there's a lot of
is that
rumors and misnomers about the vaccine. And so you really have to engage in culture.
I would have said that yes, there certainly are areas where that is the case, but in fact we've been able to, through whatever means, largely reduce, not eliminate, reduce the amount of measles. We have nearly eliminated, we have eliminated 2 out of the 3 serotypes of polio. We are probably going to be able to eliminate the other if they don't. Now, on the other hand, if you are a vaccinator in Pakistan, there's a fairly good chance you're going to get shot and killed. So there are side effects to vaccination. But I think most of the world accepts it enough that we were able to make those kind of successful interventions. It's how we got rid of smallpox. I mean, so it can be done. The opposition is not so much so that we can't make those nearly complete public health programs successful.
I guess just to kind of came out, I would say that we have to approach it culturally and be culturally appropriate.
That seems reasonable.
I mean, because the communications piece in West Africa was significant.
And if you bring in an imperial perspective, this is the right thing to do. We're doing it in North America. That's not the way to persuade. It has to the people who are of must be the ones who are persuaded. Yeah, I agree. Okay, well, thank you very much.
So, Dr. McKinney was not only inspiring and brain-provoking, but he actually made a really great introduction to our next speaker, because this is somebody who did act. So Dr. Ritz Alou from GSK was, I think, one of the first people who sort of sounded the alarm that there did appear to be an outbreak of Ebola, despite the unwillingness at the time of the WHO to declare it an emergency situation. And Dr. Alou is a vice president and head of GSK Global Vaccines. He's now in the R&D Center in Rockville, Maryland. Prior to this, he was VP and head of clinical research and translational science, vaccine discovery and development of GSK vaccines in Witzenard, Belgium. Previously, he served as deputy director for vaccines and infectious diseases, development of global health for the Bill and Melinda Gates Foundation. He's an expert in vaccine development and has worked in this field for more than 30 years. He trained in internal medicine and infectious disease and began his work on vaccines at the Walter Reed Army Institute of Research, where he led the team that with GlaxoSmithKline developed RTS as the world's most advanced malaria vaccine. Dr. Ballou has numerous publications in the field of vaccine development and infectious disease. So,
So, Julie, thank you very much for inviting me in. But that was a great talk, and coming from sort of a very intellectual view, we're going to talk about some of the practical things around what happened. And I want to correct, I didn't pull the alarm on the outbreak. I did talk to WHO very early on about the possibility of using a vaccine to protect healthcare workers. And this was the driver of why we got involved in it. And it turns out to be a lot of serendipity, as it always happens in this field. If you're not prepared, you're not able to act. But I want to talk a little bit about what we did. And then the second half of my talk is about some reflections on what we think we could be doing better. And a very specific proposal that GSK has been in discussions with a number of governments and agencies. So it's a little bit to tell you about GSK vaccines. I mean, our strategy is that we will lead our industry, the vaccine industry, to help improve health globally by continuously delivering better vaccines, protecting more people, and to run our business sustainably. And that last piece is important. We cannot do things that we want to do if we don't have a sustainable business. And having a sustainable business means that we do have to pay attention to shareholders, and we do have to be able to guarantee, by doing this, we guarantee the supply of the vaccines that help people around the world. And this is not only true for GSK, it's true across the industry. And when we do things like focus on epidemic outbreaks and put resources on this, we begin to cut away at the sustainability of our organization. And this is why we really need to think about what we're doing. We are one of the major providers of vaccines in the world. Every day we deliver 2.5 million doses of vaccines in more than 90 diverse countries. We produce vaccines for more than 90% of the diseases for which vaccination is recommended, and 80% of our vaccines go to the developing world by volume. So it's a very substantial footprint that we have, and we take our role in providing vaccines and supporting healthcare very seriously. Now, we talk about partnership. I looked at this poster over here, and you guys forgot a couple of partners that we had to get involved with in this Ebola outbreak. Everybody listed on this slide was a key player in our program, and we did have to develop contracts, agreements, all of these things that we talked about, the amount of work that went into this. It wasn't an army, but it was a small group of lawyers who kept very, very busy around the clock. We had one in Alaska, several in Belgium, and I don't think the lawyers ever went to sleep. It was all going on in the background while we were trying to actually do the work. And thanks to the help of all of these groups, we were actually able to move a potential vaccine candidate forward in the context of an extremely unusual situation. Just to recap the timelines on this outbreak, people are aware of this. The first case was probably early December 2013, but it really wasn't until about 6 months later that the outbreak there was an outbreak was announced by WHO. And it was, I read this on the 23rd on the internet, essentially. You've got this issue in West Africa, and I'm very into infectious diseases. I have worked in Africa, and I realized that where this outbreak was occurring in cities, in a pretty populous, a lot of transportation routes, a lot of communication was very different than where we had seen Ebola before, and therefore the potential for this really getting out of control was quite real, and that's what led me to contact WHO and begin this dialogue. It took the WHO some time. When I first talked to them, they had no policy and no thoughts about how we might use a vaccine. The idea was, well, if there's no licensed vaccine, there's nothing we're going to do. We happen to have come into a potential vaccine by virtue of an acquisition of a small Italian biotech that had been working with the NIH to develop an Ebola vaccine, and they had just so happened to have produced some clinical lots of the very strain that was actually circulating in West Africa. So there was this potential to say, we have Ebola vaccine, it's never been in the clinic before, but could we accelerate that process with initially a focus on helping to protect healthcare workers? I remind you, more than 800 healthcare workers, a lot of these physicians, nurses, people that are actually taking care of patients became infected, and more than 530 of those died. So and that essentially crippled the ability of West African health system to actually manage the epidemic. So this was really sort of the incentive of my engagement in this. It took the WHO some time to integrate how would we take experimental vaccines, experimental medicines in the context of an epidemic. And of course, at the time that this PHIC was public health emergency of international concern was announced, we had very dire predictions coming from the London School that if things were to continue like we see on this trajectory, by Easter of 2015, we would have depopulation of West Africa. Now, that was a very sobering statement by very smart epidemiologists, and I think that on the one hand, this comes to the tension that you talked about in the ethics. People were faced with, oh my God, depopulation versus experimental medicines, and that tension was palpable in the room at WHO. Anyway, we were able to, the previous slide, pull together a consortium, and I had spent all of my career working in malaria, and there are already a lot of established contacts in sub-Saharan Africa and with investigators in both Europe and the United States. And it was a natural inclination to pick up the phone and call people that I knew, who, by the way, could work in West Africa. And we were able to work with Wintiles, who had the capability of putting clinical operations activities in West Africa. So it was serendipity, being prepared by virtue of other work that we had done, that we were able to cobble together a team that was totally committed to doing something, but of course then we had to put all the legal agreements and the contracts in place, because this is, as you said, a very regulated industry, and we just can't do things without having the proper agreements and things in place. So the GSK program was one of several that followed it. Swati just came in, I think. The Merck vaccine, the GSK vaccine, and the group from J&J were three of the groups that were able to drive candidate vaccines into the clinic. The Merck vaccine was actually able to make it through a phase 3, a very unusual and creative phase 3 trial. We have been able to get through phase 2, and Merck and J&J is essentially in a small phase 2 status right now. We're able to do some additional work, but in the absence of ongoing transmission of any significant amount, the ability to demonstrate efficacy beyond this is really limited. And I think that, just reflecting again on some of the dialogue around this tension between potential depopulation and what do we do, there was a very interesting debate about study design and can we use placebo-controlled trials. And it was a very polarizing argument, but I think it led to the clinical trial experts to go back and seriously rethink how we might get robust data in the context of this situation, and I'm sure somebody's going to talk about that in terms of the trial that was done, the vaccination trial, which I think really has helped move the field along. And it came out of that tension between, what can we do and what should we do. So just a few words on our vaccine. It's based upon an attenuated chimpanzee adenovirus that we take out essentially part of the adenovirus gene that expresses adenovirus surface proteins and replace it with a piece of the Ebola vaccine, of the Ebola virus, so that when it infects cells, it now causes those cells to express the Ebola glycoprotein on the surface, and we make an immune response. That's in essence how live attenuated vaccines work. The Merck vaccine uses a different vector, but a very similar approach. Our vaccine is non-replicating. The Merck vaccine is replicating. The price you pay for a non-replicating vaccine is that you have to give a very high dose, because what you give is all you get. In contrast, in a replicating vaccine, you can give a much lower dose because it replicates in the body. There may be trade-offs in terms of safety. Fortunately, for both vaccines, they appear to be quite safe. But when you're starting off and you've never been in the clinic before, these are really important issues to consider in terms of thinking how you pace at which you move forward. We started our program in September of 2014, and manufacturing internally is still ongoing. We had made a commitment, we're still doing this, that we would produce a large bulk of this vaccine that could be potentially available in the event that the public health agencies want it. It uses modern technology. It is a very complex process, and because of the rapid development of it, all the characteristics of the vaccine we would like to have, such as thermal stability, we were not able to do that at the same time that we were developing the vaccine. So we do have a product that does require -60-degree storage, although it is stable at more conventional temperatures when we're actually working in the field. And in practice, it hasn't been a problem. The vaccine was immunogenic. I said that there was a cost to a non-replicating vaccine. And what we found is that we had to give the highest dose that we could physically manufacture. So this puts real constraints on our ability to generate rapidly the number of doses that we requested. And it was one of the main reasons why we were really unable to be ready for the phase 3 trials when they started, because we just could not supply enough doses at the time that was requested. And again, it's this tension between a moving epidemic and the work that has to be done. We conducted 5 phase 1 trials. There was a trial in the United States, a trial in Oxford, a trial in Switzerland, several trials in Oxford, and a trial in Mali. And we found in Mali, which is the place where we did the malaria vaccine trials, was a great place to do vaccine. It is a great place to do vaccine studies. There's good leadership there on the ground, and the community understands what vaccines are about. We've had really no issue in terms of acceptance of people being involved. And we found that the vaccine was immunogenic and was well tolerated. There were no safety issues, even at the very highest doses that we tested. We did confirm that we need this high dose, and we still have about 70 people that we've had to pull off of other projects working full-time on the manufacturing of this. And this has put a constraint on our ability to develop other important vaccines. We were asked to participate in a phase 3 study called Prevail that was run in Liberia under a joint relationship between the NIH and the Liberian government. This trial was launched as a 3-arm study, so the vis-a-vis vaccine from Merck, the GSK Chad 3 vaccine, and a placebo control. So we were able to have one of the optimal study designs of a placebo randomized controlled trial put in place, but shortly into the trial, essentially the epidemic in Liberia collapsed, and the phase 3 design was converted to a phase 2, and no efficacy data were able to be generated, although we did get safety and immunogenicity data. We are continuing a large phase 2 program. It's being conducted in 5 countries across countries across Western Africa, Mali, Senegal, Cameroon, and Nigeria. These are countries that were adjacent to the epidemic area. We felt that these were countries where we could do carefully controlled phase 2 studies and collect the safety and immunogenicity data that could support a regulatory file. The work doing these kinds of studies within the 3 target Ebola countries would have been very, very difficult because the infrastructure was essentially collapsed there, and our ability to generate really high quality data on safety, immunogenicity, co-administration, those kinds of things were limited in that area. There is a proposed licensure pathway. We worked very hard with WHO and the EMA and FDA about various pathways. We're in discussions with them. We don't know whether there is going to be a licensure, at the end of the day, a licensure pathway for our vaccine, since it was not able to complete a phase 3 efficacy study with disease. There are other potential avenues, but they are quite complex, and it's premature for us to be able to conclude that we're going to be able to do this. But if it is possible, we certainly intend to follow through on this. So how can we be better prepared to tackle emerging threats such as Ebola, Zika, rulers, etc.? The impetus for us to think about this really came out of our Ebola experience. And we got involved in this because we felt we had a platform technology, this adenovirus platform, that was amenable to targeting this particular disease. It was, in theory, a more rapid response type thing than some of our traditional alternative ways of making vaccines. But this response is very disruptive, and the delays to core R&D activities may result in greater morbidity and mortality than the health crisis itself. We delayed RSV, we delayed DINK, we delayed other programs for which we know these are ongoing common diseases that people die of. And GSK and other companies possess proven technology platforms that could provide solutions. So we propose to make available through a separate organization, and we're calling for one of anything else to call it BPO, Bioproduction Organization, a facility and a platform, a way of working that would use agreed upon platform technologies that GSK would provide. We have facilities available in Rockville, Maryland that includes 2 pilot production lines. It's the building where I'm building our new RDC. It has an existing facility in place. It is GMP capable. It's in a mothballed state right now that could be adapted for use. And we have all of the required expertise in vaccine development to support an accelerated vaccine development program. We're not seeking profits to the BPO, but we are working with governments and funding agencies to cover operating costs and provide appropriate liability protection. In fact, in practice, the vaccine targets and platforms would be recommended by an international governance board, WHO and others. There would be a scientific advisory board that would help us decide, are we working on the right target with the right platform, and have this transparent and of course all the people that would contribute to BPO would have the ability to weigh in on decision making. The proposal would be to take vaccine development for selected targets through early phase 2 dose selection, so that this implies pilot manufacture at a reasonable scale, so that we would potentially, after doing the phase 2 studies, we know what the dose is to give, we know what the immune profile looks like, and if that's promising, we could take the remaining lots of material, freeze them down, and they could be potentially available for use on a much faster track than having to do what we did before, which is starting from scratch. This has been discussed with governments and NGOs with quite an enthusiastic response. We have the U.S. government, BARD, and WHO have issued relevant proposals, and we're really in advanced dialogue with them right now. What would we work on? Well, there has been a priority pathway that has been developed. The list of known threats is quite long. There's more than 75 pathogens, but there's a list of about 8 plus 3 that are considered to be severe epidemic threats by the WHO, and for which these would likely be some of the first things that we would work on. Obviously, Zika was not on the list at the time because we hadn't heard of Zika before, really. few experts in played virologists knew what Zika was, but most people didn't. But clearly it's an example of that. If BPO had been in place 2 years ago, this is where we would be working on Zika. But as it is, this is the timing is great. Just to give you a sense of what we're doing here, this picture here, sort of antibody looking like building is the facility where we are. Wing C is essentially a manufacturing facility and has where we position the DPO. The rest of about 3/4 of the rest of that is going to be our GSK's U.S. vaccine headquarters. We're in the process now of renovating and as you will see on the last slide, hiring. But we will have the ability to do platform selection, antigen design, technical development, tech transfer, and we would focus on a couple of platforms that we think are particularly suitable for rapid response. As I said, there are 2 pilot plants here that we can be run in parallel. We would have the ability to do GMP analytics, QA, raw material stability, clinical immunology, and administration. So a full functional organization distinct from the vaccines organization of the co-localized. So we could share things like regulatory things, so that sort of stuff. This is a list of some of the technologies that we could potentially contribute to BPO, but I want to focus on a particular, just one as an example. It's called self-amplifying messenger RNA. It's a brand new technology referred to as SAM. This methodology bypasses, because it, as you'll see, you don't get an immune response to the vector, and it has a demonstrated ability to provide a rapid response. We're talking about in weeks to a new pathogen. And the way this works, it's a disruptive approach. What you see in the picture on the left is an RNA viral particle, and an RNA virus is coated with proteins that direct an immune response against it, and that's one of the things that limits the ability to reuse an RNA vector like this or another live vector, because you make an immune response against the vector. But the trick here was essentially taking the guts out of this virus, the part of this that has the machine for replication, and then in there you can insert your vaccine target, and you then mix this with a delivery system that allows this material to get intracellular. And since it is self-replicating, the messenger RNA replicates in the cytoplasm of the cell, generates the protein of interest, which then is expressed and you can get an immune response. The idea here is that if you had a new virus or a new pathogen, you have a sentinel organization that's already been put in place, really being driven by the experience with pandemic influenza, monitoring outbreaks, where information is fed through the internet, and this system is working good now. The idea then would be that you would be able to synthesize the gene, produce the product, and have, by virtue of pre-stockpile vehicle and rapid ability to manufacture this messenger RNA vaccine component, a vaccine in a relatively short period of time, using a very standardized platform that, in theory, this has to be discussed with regulators. Once you've done this a few times, now the process becomes the vaccine, and reinsertion of a new gene in there becomes a minor variation in that process. And the feasibility that we can respond to has already been demonstrated and published in the case of H7N9 and avian influenza. The time from release of sequence data to confirmation of H7 expression in the cell line using this took 8 days. So this is a very fast response. This has not yet gotten into the clinic, but we'll be doing that this year. We think this is a very potential, as an example, and there are other approaches that other companies have, that the ability to respond quickly to an emerging threat it's something we can actually talk about and actually do it. And this was not the case several years ago, but we think that this needs to be done outside of the business as usual part of our industry, because it is so disruptive. So in conclusion, Ebola was a global wake-up call. The work to prevent another Ebola epidemic is far from complete. We don't have any licensed Ebola vaccines, and there are going to be other Ebola outbreaks. responding to similar threats in the future requires a serious rethinking on the part of the global community. I am totally convinced that industry will be a necessary part of the solution. Yes, we will work with academics, we will work with, in partnership with many people, but if you're going to deliver a vaccine, you have to have industry involved. But industry cannot take this on alone. Governments and global health authorities will need to take a leadership role in this. Access of new vaccine drugs and diagnostic to communities that need them has to be an integral part of the proposed solution. We've made several bold proposals, including our BPO, the IP, and data sharing that we talked about earlier today. And we are very much looking to engage. And I think the work that Julia and you and your team are doing in terms of how do you put these partnerships together, how do you make this work, is really going to help us in that. So thank you very much.
Questions? I'm sure there are. Go ahead.
I think it's really cool to have extra manufacturing capacity for emergencies. Can you give us any idea of the cost of the facilities and also about your criteria for choosing which vaccines would get used in the facility?
Well, let me go to the second part of this. I mean, the choice of what target to work on and what platform to work on would be really the work of the governance board, which will involve, if this gets put in place, will certainly involve players like the WHO. So this has to be informed by what we think the most likely next threat is going to be. When I talk to folks at BARDA and WHO, what's on high on everybody's list right now is MERS, as an example. Okay, highly pathogenic, with the potential for human-to-human transmission. So there are going to be criteria around what it is that what target is likely to be most threatening from an academic point of view. Then there needs to be a scientific analysis of which of the platform technologies is best suited to attack that particular antigen. It turns out that most of these things turn out to be viruses. So we have sort of biased our proposed selection to viruses, but we have capabilities to work for some very interesting technologies against bacteria as well. So and toxins if that was a potential threat. So there are different approaches. We certainly need to build out what that scientific advisory group looks like. What is its membership? What is its expertise? What is the governance process? That tells us what to work on. The cost of, there is an initial investment that needs to go into the facility to take what is a current set of GMP facilities that were designed primarily for cell culture, monoclonal antibody type of production, and to replace that equipment with modular equipment that's more aligned for vaccine development. So there is an initial cost. It is in the tens of millions to do that, but there's that piece of work, and we think that would take about 18 months to get that to the point where we were actually able to put something out the other end. And then there's ongoing requirement to fund staffing up. We think the whole organization would end up with about 120 employees within this. These would be GSK employees, but working in separate organizations. So it's a substantial budget.
Yes.
Well, yeah, just so you mentioned attracting and taking advantage of technologies from other companies, so bringing in the platform technologies that other companies have for vaccine development, sort of parallel, I guess, to GSK. How would that be done, both from like a development perspective as well as just physically in terms of?
Well, we are not suggesting that other companies come and work in. So we think the BPO concept could be replicated. Now, that's not to say that we could have put in place the agreements that would allow a Merck, for example, to put a platform technology in this BPO. But to date, we haven't taken the discussion that far. But certainly we have invited the larger vaccine community to think about this as a strategy.
This question of platform technology scaled up. GHIA, which is the company that GSK acquired and scaled up for Ebola, was that sort of an acquisition that was serendipitous, or was there a larger foresight in that?
No, we bought Kairos for the adenovirus platform, and we're using it for RSV. That was the primary reason. As I started to dig through the portfolio that came with that, which was quite extensive, and Kairos' operating model was find as many collaborators as we can, give them our vaccine, let them do studies, and something's going to work. And in fact, that's how we came into our attention. Buried in that list of activities was an Ebola vaccine. And they had very compelling data that they could completely protect against a highly lethal challenge in primates. So when I saw that, I said, okay, this is something we could potentially do. So it was serendipitous.
So we're very supportive of a more rapid way to make vaccines. But I'd like to hear your thoughts on a couple of issues that we always run up against. So I understand the utility of using genetic information as opposed to trying to get in on strains, because sample sharing is always a major issue in these outbreaks, which is not a scientific or technical issue, it's problems in themselves. So the first question is strain selection. How that's deciding on what strain sequence to use is a major decision point, and basing it on genetic data in the midst of an emerging outbreak may be fraught with risk. The next problem we run into is testing the vaccine preclinically. If you don't have a lot of human disease pathogenesis that you understand, how do you identify the right animal model with which to know whether or not the vaccine works? And then the other issue is, how do you decide what immune response you actually need to generate in order to have an effective vaccine when you just don't have a lot of example of the disease?
These are great questions, and of course, these are exactly the questions that face any vaccine developer at the beginning of a vaccine development program. And I think in the case of Zika, every one of those points is absolutely spot on. Zika we may not be ready for a Zika vaccine because we simply do not understand enough about it. I think there are other pathogens on the list that we do have a better sense of. For example, MERS. I think there's pretty good demonstration that spike protein on MERS can protect in a challenge model in animals that appear to be relevant. But MERS is not a commercially viable target, and it's why none of the big players is working on a MERS vaccine with any kind of serious. But if this thing spread, we would be the first ones to say, why aren't you working on this? But this is why you have to have a great scientific advisory board, and you have to think through very carefully all those key decision points, because if you go down the wrong track, you're wasting your time. And it's a gamble, so there's a risk, there's always risk.
I'd ask a sort of broader macro question. So, Mary Moran and Policy Cures estimate is about $3 billion a year now. Global Health R&D. WHO Consultative Expert Working Group for R&D Financing says we need at least $6 billion a year. That's probably a conservative estimate. Let's assume that donors, and I spend a lot of my time working with donors trying to persuade them of the need to spend more. Let's say there's an additional $3 billion. What are the sort of incentives to get more GSKs and Genentechs and Merck's and other companies to the table for this kind of work?
Well,
what have you learned from your experience?
I think the incentive for companies to do this is to have gone through something like this Ebola. I mean, I'll be very interested to hear what Swati has to say about Merck's experience. It is very, very disruptive, and we can see the impact inside the organization very clearly on our timelines, on our ability to manage multiple things. We have used up all of our excess capacity to be able to respond. That is our incentive. And this was not our first time doing this. The same thing happened with pandemic influenza. Now, it happened to be that GSK had an influenza platform that was amenable, but it was hugely disruptive to the organization. And if we consciously said, we will investigate our SAM platform in collaboration with the NIH as a possible approach to Zika, but we're not going to take on an internal Zika program like we did with Ebola. There are too many uncertainties, and we just simply do not have the bandwidth.
So what's the plus side of your company that you could say to other companies, look, it was great for us because of X?
I mean, this is, why do we think we should do this? Because we think it's necessary. The world is going to continue to be threatened with epidemic disease. Vaccines are a proven way of interfering with these, and we think that as vaccine manufacturers, and I'm going to use an ethical thing, we have a moral obligation to be part of the solution. We don't want to be making the problem worse, and it's this tension that you talked about very elegantly, but we are trying to do the right thing. And it's as simple as that.
So, for Barry Morgan and the execs, and I'm going to thank you very much for doing my talk for us. When we look at this issue, one of the values to a company is, especially with platform technology, is your internal company gets more experience with that platform technology. So, the Osiris RSV project, to the extent that there is investment by the company in learning how to manufacture large amounts of virus, that will bleed over into the commercial elements of the program. Regulatory experience with the platform. All these things are
That's true.
less monetary, but are of value to a company.
But those are things we would have done anyway, but we certainly will, we have learned things from this and it will be useful. Okay?
So after you mentioned the influenza outbreak was a major learning lesson for companies worldwide, and one of the things that came out of it was a new virus sharing framework. But that hasn't been adopted for other viruses such as Zika, which is a problem with sample sharing right now. Is there anything GSK or other companies are doing to get around this issue of data sharing, specifically with biological?
I think this is something that has to be managed above the level of the company. This is a very serious issue. It does need to be solved, but I don't think companies can solve this. We can be partners in this, but there's an access implication, and that certainly is we'll talk about that. So,
Right now.
Let me get off. Thank you very much.
Thank you so much. Couldn't have been a better lead-in.
While you're introducing me, I'm going to.
You gonna introduce yourself?
No, I'm going to do
You're gonna do this. All right, I'll say it. Right. So
What's the introducing me?
that's good. And you can that's So Dick Wilder was previously the Associate General Counsel for IT Policy at Microsoft, but now he's the Associate General Counsel for the Bill and Melinda Gates Foundation. He has an emphasis on intellectual property, especially on global patent issues. And formerly he was a partner at Civic Austin, where he counseled clients in shaping policy positions and strategic initiatives, including global health, international disputes, and technology. At the Gates Foundation, Mr. Wilder is responsible for addressing legal issues that arise in connection with the development of interventions in the field of global health, including drugs, vaccines, and diagnostics. He holds foundation-wide responsibility for management of intellectual property and addressing issues relating to access to data. And I refer to him as the holder of most agreement database for all of the global health agreements. So he's been very helpful to us in sharing some of their provisions and how we might improve things. And so I'll let you begin.
Yeah, thanks to it. So I really appreciate the opportunity to be here and to have this conversation, basically. And thus far this morning we've had some really interesting presentations, and I've got some notes because as I go through mine, I'm trying to make reference to some things that have already been said. At the Gates Foundation, I don't really have a slide set that talks about the foundation as such, but I think most people in the room know of the Gates Foundation, the fact that we provide a significant amount of funding for research, development from early stage discovery, all the way through clinical trials and putting new products into the market. I'm going to be talking about some of the legal issues that arise in connection with that activity. What I wanted to do is to maybe start where Rick left off, and that is reference to global access, because for what we do, it's one of the threads that runs through all of our activities, and it's a concept that was put in place at the foundation over a decade ago now. And what we want to do is to be sure that as we fund our activities, whether they are through our grant-making activities, contracts, program-related investments, or we're increasingly making equity investments, for example, in small companies, that our global access objectives are met. And they basically have two prongs to them, if you will. One is that the information and data that arises from our funding is made broadly and rapidly accessible. The second piece of it has to do with the product access issues. That is that if we are successful from a scientific and technical perspective in bringing products through the development process and into the market, we want to see to it that they are available, including on price terms, to our target populations, which are largely the poor people in poor countries. And so that's really, like I said, the thread that runs through our activities. Now let me talk just briefly about our engagement on Ebola, because it's slightly different from what's been discussed thus far, especially when it comes to what Rick just described around the activities of GSK. Ebola and Zika both are not and were not diseases that fell within our normal programmatic activities. So, as I said, we have funding, we're engaged a lot in research and development, or funding research and development in infectious disease. We have established programs dealing with enteric diarrheal diseases, with HIV, with malaria, with TB, with neglected tropical diseases generally. And those diseases weren't ones that fit within, or were planned to fit within existing programs. And so in that sense, it arose rather rapidly at the time that the Ebola crisis hit. And I think it's been described thus far in terms of what the timeline was for the Ebola crisis. That said, we did recognize within the foundation that we have some capabilities that are at least additive, and in some cases actually special and differential from those capabilities that others have in the global health arena. One of the capabilities that we have is the fact that we're basically run by two people, Bill and Linda Gates. And so if they decide they want to do something, we do it. In this case, when it was presented from staff internally, a proposal that we should actually play a role in Ebola, it was decided within like 48 hours from initial planning to the presentation to the ultimate decision to allocate $50 million to do work in Ebola. But as you can see from this slide, we identified a number of different pillars, if you will, of the activities that we could do in order to take advantage of that $50 million commitment. A lot of it, quite frankly, in terms of what we actually did, had to do more with pillars 1 and 3 than the others, and that was, we were in a position where there were, actors that were on the ground and engaged in addressing the Ebola crisis, like Médecins Sans Frontières, for example, and others that were the frontline providers of services to attack and deal with the crisis on the ground in West Africa. And so what we could do is, for those organizations, we'd be able to provide very rapid funding in order to enable them to get what they needed in terms of resources on the ground and to be able to bring in resources to support their efforts. Recognizing that what we were able to do was to be nimble and very quick with respect to our funding, and that would then complement what others were able to do that would take a bit more of a timeline, but they would be able to commit much greater resources to the effort. There were some ongoing research and development activities, including the vaccines that Rick described and others, that we were able to basically complement and sort of almost piggyback, if you will, on top of what others have been doing. But again, the notion there being that we could be nimble and quite quick in terms of addressing that. And so this is kind of representative of what we can do in an emergency situation in terms of taking advantage of certainly our funding, which is considerable, but then the nimbleness in which we can respond. But just to emphasize that much of what was done with respect to the Ebola crisis, required much greater resources than we would have been able to have, that we were able to provide in terms of what was done in terms of addressing the crisis on the ground. What I wanted to do, and I've got a couple of slides on this theme, and what I wanted to do is to kind of expand the discussion a little bit more, maybe take it into a slightly different direction and talk about some of the legal issues and agreements and so forth that arise in the first instance really with respect to any activity related to the development of new drugs, vaccines, and diagnostics, and then offer some thoughts as to how those those same activities play out in the context of an emergency situation. So as I mentioned before, we have our global access obligations, and here I'm kind of generalizing it. Obviously I know from the foundation's perspective what that means and how we implement that in the agreements that we do. But I think it's a concept that we can think of as more generalizable in the sense that in the end, when we fund, when we're all involved, anyone in this room actually involved in activities around the development of drugs and vaccines, we do want to see to it that it actually has the intended impact on the populations that we know are going to be affected or are affected by infectious disease. And often this concept of access and global access tends to get reduced down to a single dimension, and often it's around the issue of intellectual property. But in terms of the work that we do, that certainly does play a role, and it's certainly one of the legal issues that needs to be addressed and negotiated in the agreements that we put in place. But there's also a host of other issues involved, from early stage discovery all the way through clinical trials, manufacturing into the marketplace. And so, in that context, we need to understand how that's playing out in those other areas in terms of regulatory approvals, what the local infrastructure is in order to accommodate the rollout, utilization, diagnosis and treatment using these new therapies, pricing, and so on. And so here, what I wanted to do is to lay out sort of a timeline rather in the development process, and then the different legal arrangements that arise underneath that. Actually, one thing that I made a note of it and I forgot to talk about in connection with the foundation's involvement in Ebola, much like what Rick was describing in terms of the commitment of resources being separate and different from what has to be done on a regular basis by the company, because you've got your ongoing activities. It was the same as I described with respect to the foundation. This was not something, either Ebola or Zika, something that was in the context of any of our normal activities, and so it arose up. And even though our involvement was relatively small, it did take an enormous amount of time from staff internally to deal with this. And so there is this notion of, and I'm going to return to this theme in a moment, but there is this notion of when these emergency situations arise, it means to a certain extent you have to set aside work that you're doing already, and to kind of put it in a kind of extreme sense. If you're working on Ebola, then you're taking time away from the teams working on malaria, so malaria drug research and vaccine and so forth, which then delays the work that's being done in connection with those other areas and those other diseases. So there is a cost associated with it, which suggests what Rick was suggesting with the BPO, and that is to have some kind of an institution, some kind of basic infrastructure, that could be taken advantage of, when these crisis situations arise, which are likely to happen more as we go forward. But to return then to this theme of what's involved is that what I have up here on the screen in terms of the various steps that are involved in product development life cycle for drugs and vaccines largely, and then the various agreements that need to be addressed under each of these stages, I think is something that is pretty kind of plain vanilla, if you will. These are kind of typical things that need to be done. So you need to have, and there was some discussion about access to materials and being able to exchange materials in connection with research activities. There's the ongoing need for data that is descriptive of the sequences for viruses, for example, as was mentioned. There's an ongoing need for data in connection with the scientific work that's done, the clinical work that's done. And so being able to define what the data and material are that are necessary for the work to be done and how you're going to achieve access broadly amongst all the participants is important. And then from a very early stage, from our perspective, again, we're looking at this in terms of global access. We want to, to basically define that endpoint in terms of what we want to achieve in terms of the resultant product being available to the poor people in poor countries at an appropriate cost. That we need to have that defined from the beginning, and that is a thread that really runs through all of these agreements. Obviously, very early stage, it's not possible to talk about price and volume and those kinds of commitments because you don't know if the science will be successful, let alone whether it's going to be able to be instantiated in a product and brought to market and so forth. You don't even know what the good is, let alone the various elements that go into the cost of goods. But as you get forward to closer bringing your product into the market, you can be more specific about it. Now, getting to the point of collaboration that amongst the different actors and different players, there's been a fair amount of discussion about, Ross mentioned it, and Artie and Rip about the fact that there are a number of different, not just actors that are involved in this process, but different types of actors that are involved. And so you have, obviously, industry is an important component, government labs are an important component, universities are an important component, and each of them has their own, motivations, each of them has their own constraints or rules that govern what they can and cannot do. And so all of that has to be factored in. And what we've found to be successful in that connection, and we've been able to do it in the context of a number of different specific initiatives that we have underway, is to have very purposeful discussions about what's necessary from really a scientific and technical perspective in order for the work to be successful, to do, again, whether it's early stage and through clinical trials and bringing a product into the market, and then using that as a roadmap for a discussion about then what needs to be included in these various types of arrangements that are put in place. So, a couple of examples from the Foundation's perspective in that regard. One is an initiative that just celebrated its 10th anniversary. It's the Collaboration for AIDS Vaccine Discovery that includes with it a number of different, three different structures really in terms of how the collaboration works. One of the structures or sets of structures is the different approaches that are taken to develop an AIDS vaccine, so from different scientific approaches. The other is that there's a set of service providers that were stood up for that process, one of which actually is specifically tasked with managing the data flows and data access in connection with the collaboration, and another one has to do with being a material repository. And then another piece to it that was innovative at the time and something that we continue to use and continue to iterate on is the data and material sharing agreement, which is a set of rules that are embodied in a legal instrument that define how this collaboration is going to work together, allocating different responsibilities from a technical perspective, but then also agreeing as to what's going to be done with respect to the sharing of data and materials, hence the data and material sharing agreement. The interesting thing about that process, and this was done like in 2004 or something, I was involved in that when I was outside counsel to the Gates Foundation, was that it was an effort to first define from a scientific perspective what was necessary, and then second to embody that in legal language in order to facilitate that. And as you all know, in connection with these issues around intellectual property ownership and licensing, data ownership, access, and so on, and the sharing of materials, is that there's every one of the actors has their preset ideas that they bring to the table in terms of how they want that to be managed. And as we put this together, we designed it in such a way that we were able to collapse those various requirements down into single ones. I would say an outstanding problem from the result that we achieved there, and something that we continue, as I said, to iterate on as we develop more of these collaborations, has to do with some of the complexity and time lag that's built into the arrangement. So, from an academic perspective, the academics, they want to be able to have a certain period of time of what you might call academic exclusivity to be able to study data sets and decide what they're going to publish on. And so, that delays access to data. Both academic institutions and industry, want to have a certain time lag involved for that purpose as well, to decide what intellectual property rights they may decide to apply for, patents in particular. And then the same when it comes to the publication of results or dissemination of data publicly, that there needs to be a period of delay in order to accommodate those requirements. And as we've gone forward with the same basic model in things like the TB vaccine accelerator, we have a TB drug alliance as well, our TB drug accelerating mechanism, that we've continued to iterate on those in order to bring the time frames shorter and to be able to move things forward more rapidly. One of the challenges, and this is, I'm sure, coming out of the work that Julie and your students are doing, is trying to figure out ways in which again those time frames can be collapsed, and in certain instances that you can actually remove some of the basic restrictions on access to data and sharing of materials. What I think, well, two things. One is that I think that we can continue to, we being the foundation and other partners, can continue to iterate on this in order to to facilitate more rapid access to data and materials. I think that part of it too, has been improved by the changed realization, I think, of a number of actors, and it's not just industry, but it's universities as well, in terms of the role that intellectual property plays in connection with the work that we're doing. And there was mention made of the recent decision by GSK to forego patents in certain countries and to manage IP in a different way through different mechanisms. I think there's been a lot of progress in that regard that frees up the ability for us to really shorten and compress not just the time frames, but also the scope of what people are interested in securing in terms of exclusivity. The other piece of that, and this, since we're at Duke University, it'd be interesting to have further discussions on this from an academic perspective, is that one of the activities that we've been engaged in over the last 2 years now is to promote open access to data and to published materials that arise from our funding. So we have a new open access policy. And in connection with that, that has to do with open access requirements around articles that are published in peer-reviewed journals, and we want them to be published and available to open immediately upon publication. Therefore, there's no embargo period with respect to that publication. And then also being published under a just an attribution-only license, so a CC BY license under Creative Commons. So not a license that restricts use in any way, whether it's for academic or commercial uses. And in that discussion, just as industry has interest and concerns about protecting intellectual property, and there has been a lot of progress over the years in terms of, not so much, well, I would say not at all, taking away the IP system or the incentive, but recognizing in a more granular way the role it plays for different types of products in different markets. The same thing on the academic side in terms of wanting to be able to publish in certain publications, wanting to be able to publish, or for the publishers themselves, wanting to have this period of exclusivity, if you will, behind a paywall before that information is disseminated, including the underlying data, is something that we continue to drive on. But the point there is that there's incentives that the academic sector has, that we need to engage in more directly, just as there are incentives that affect industry, that we need to engage in more directly. I think the last thing that I wanted to talk about, and this again is from Foundation's perspective, would be the different types of investment vehicles that we use. The different type of investment vehicles that we use at the Foundation, and how this concept of global access and IP management, data access, material access plays itself out in these different arrangements. So, and again, this is Foundation perspective, or Foundation specific rather, but I think I can make it more generalizable here. So, historically at the Gates Foundation, our funding has been through grant arrangements. And, in the early days, the number of grants was relatively small, but the size was larger than it is today. And so now we're making more grants, but they are smaller ones, and we have more direct engagement with our grantees as a consequence. We're also making more grants to entities in the for-profit sector. But in connection with grants, we typically will allow the grantees then to resolve what they're going to do around intellectual property itself, just as legal instrument. Are they going to seek patent protection? If so, where they're going to seek patent protection, and for what subject matter, and so on. So there's a fair amount of liberty that's allowed in that connection. The only thing we're interested in is not so much exactly the existence of the IP rights, but how they're going to be exercised in order to achieve our global access objectives as I described. In the case of contracts, which we do with respect to materials that are produced for our internal use, we'll own the intellectual property, which is usually just the copyright in a white paper, study memo, that type of thing. We're investing more in developing new software solutions, especially in the education space, and so for that, we typically have more complex arrangements around intellectual property ownership and licensing. And then program-related investments. And this is one where it perhaps intersects most with the kind of BPO arrangement that Rick was alluding to, or talking about, arising out of GSK. And this is where we'll make an investment in an entity. Often it's a company, it could be an investment in a nonprofit as well, but let's just take it from the perspective of investments in companies, where we'll, sometimes we'll make a loan to enable a company to increase the manufacturing capacity for a vaccine. Sometimes we'll make let me just talk about the loan mechanism. So we'll make a loan to increase manufacturing capacity. We obviously, from our perspective, we have our charitable objectives that are to be met as reflected in the global access principles. So if we make the loan to build greater capacity for manufacturing of the product, then we expect that that product then will be made available on global access terms to the markets that we aim to serve. As we make investments in for-profits, it's often smaller companies, and sometimes, most often those that have recently been spun out of a university. We're interested in that because they've got a platform technology that we think might have pretty wide application with respect to the work that we do. And in those investments, we also want to see that our global access objectives are met. And there we get into very detailed discussions around the intellectual property that's held by the company, what they anticipate to be developing going forward as a consequence of our investment, or it could be in addition investments that come from other sources. But we're interested in understanding, again, what IP exists or might come into existence, and how then that's going to be managed. And core element is to ensure that as work is being done, that there will be a certain element of the activity of that company that is directed towards achieving our global access purposes. And so, we have, like I indicated, we've got a number of different programs underway that are aimed at addressing problems with infectious disease in the developing world and developing a wide range of technologies, drugs, vaccines, diagnostics, and others as well. And so, we identify that to these companies and want them to ensure that they make available the resources that they have and continue to develop to address those problems, and we'll work with our partners in order to do that. While at the same time, recognizing that as newly minted companies, that in order for them to survive, they need to be successful from a commercial perspective. They need to be profitable in order for them to be sustainable, and ideally that they will be so that they can continue to invest in the technologies that will continue to be applicable to the problems that we've identified. And so, striking that balance from the outset, something that we spend a fair amount of time doing, and continuing to strike that balance as we manage those investments is something that we continue to do. So how does this all kind of play out in the context of emergency situations? Well, from my perspective, and again from the foundation's perspective, we've had some involvement, albeit limited, in Ebola. We're looking at some of the issues around Zika, in a sense, kind of repurposing some of the projects that we have ongoing around, talking last night about Wolbachia, gene drive, and so on, and what applicability those initiatives might have to address Zika. And as has been, I think, quite well described, in the context of an emergency situation, there is this drive to produce results and to produce results quickly. The one thing that I think that we have been able to add, and we actually started on a program along these lines in Ebola, but the Ebola crisis began to wane, as was described, and so we didn't really stand up a collaboration for AIDS vaccine discovery kind of initiative for Ebola. But there is work that can be done in order to develop a common understanding of how some of these issues should be addressed in the context of global health emergencies. For example, around the data access, IP ownership, IP licensing, publication, access to materials, and so on. And we have, as I've outlined, we have some experience in having done this in the context of other programs that have the luxury of time to be able to work out these arrangements. I think that what we need to do is to do really what Julie has been describing, the work being done here, and that is to look at those examples and see what we can do to further iterate on them to make them more suitable for emergency situations, so that you can then pre-establish the kind of arrangements that we all know are going to have to be put in place, that then can be repurposed in a particular context. The other thing that strikes me, and this is something that I've had some discussions with people at the O'Neill Center at Georgetown, because I also teach at Georgetown Law, but that one of the things that would be interesting to contemplate would be the need to have some kind of a body or some kind of a body or an ongoing initiative that brings together the legal and the business people, that have had some experience in dealing with addressing the legal issues and agreements in the context of emergency situations, and have them engaged on a regular basis. And there's kind of a group of usual suspects, as it were, that are involved in this arena, in the WHO, in U.S. government and other governments, the Gates Foundation, industry, and so on. the idea would be to take advantage of the fact that there is this core of talent that has been involved in some of these initiatives and has basically successfully solved some of these problems, and find some mechanism to keep them together so that as the new crises arise, then you can field a team of people that is capable of doing that. And maybe part of it might be taking that on board in the context of the BPO as Rick was describing, and if other companies take up that challenge to do that. It really hasn't been successfully rolled out and implemented in the context of the WHO or other international organizations, but it's something that could be contemplated. So with that, let me just stop. I know that we have some more time for questions, and hopefully there'll be some questions arising out of this when we get into some of the more details. But just to conclude from my perspective is that there's a fairly well-trodden path, I think, for a lot of what I put up on the slide in terms of the legal issues and types of agreements that need to be addressed in connection with just drug and vaccine development across the board. There's a fairly well-trodden path in terms of working out the suitable arrangements with the different actors that are involved and different types of actors and specific institutions. I think that more work can be done to generalize the results of those experiences, especially to accelerate what needs to be done in crisis situations that arise. But I also think it's important that we recognize that, as we are putting together these kinds of arrangements, it's in the end, it's people, it's lawyers, it's technical people, business people, and so maintaining that body of knowledge and will and expertise amongst that core of people that have been engaged in these kinds of activities, I think is very important. So thank you. Oh, sorry.
So you articulated, obviously, that Gates really is not in the business of responding to outbreaks. You've got your set of things that you deal with.
Yeah, except for on a limited basis.
Well, on a limited basis, understood. But one of the things that comes up is that normally when you work on a pathogen by pathogen, threat by threat basis, you're always looking for the best solution, the best technology that you can develop to make the best possible product intervene. And sometimes what you end up with is the second, third, or fourth best technology for that may be something that is not only applicable to that threat in particular that you're focused on, but they may be applicable to a wide variety of things, some of which potentially are emerging threats that we may offer us a capability for effective response. But because you're focused on a particular threat and you're trying to get the best solution, you tend to not favor that or deprioritize it. Does that consideration enter into your decision making about what sort of things you're going into, focused on in terms of technologies you want to invest in?
Yeah, when setting aside the emergency situation, which is the focus of this discussion, but when we make funding decisions about what we're going to engage in, we do. I would say that even though it's unstated, that the general rule is to look for the best, or to identify improvements that could be made on existing technologies in order to either accelerate the work or to make it more productive and effective. So that's, in a sense, the mission of the organization would be to improve the science and technology. That said, I think that there's also recognition that there's much that exists that could be taken, made better. For example, I think it was mentioned, I don't know who mentioned it earlier today, but say repurposing compounds that may have been identified for the treatment of a particular disease, but then maybe susceptible to being repurposed for something else. So that's something that we're paying more attention to. I don't know if that's answering your question, but we do look to developing new and improved technologies, and so we're always in that sense looking for the best.
So I guess, so internally within Gates, for example, actually try, there may be some common
Oh,
technology.
I
And
see
then
what, yes,
some of
absolutely.
those may actually have very broad applicability that make them extremely desirable, but they may not be the best for each individual thread that you're focused on.
Yeah, no, absolutely. So, the agreements that we have are broadly enabling in that sense, so that we, to the extent that it's possible to negotiate, we'll have, gain access to a technology in the context of a project we're doing on malaria, but that it could be then, used if there's agreement with the party and the funding is available and so on for the treatment of, TB or other diseases we're working on. So yeah, we do look at it in that sense. And the PRIs, or program-related investments, equity investments that I described, are very much engineered that way, so that we're getting access to that technology, not just for a single disease, but across the board in terms of everything that we and our partners work on. Yeah.
Can you give us an example of access provisions for specific drugs? So we're going to help you develop this drug, but you can't price above X, or it has to reach Y patients. Is it that specific for your access provision?
It does. It gets to that point, where, at the end of the process, like the meningococcal A vaccine that we funded the development of and brought to market. At the end of the day, we do work with the manufacturer, in that case with Serum Institute, work to identify what we're going to require, what is going to be required, I should say what we require, what is going to be required in order to address the needs of the market. That involves obviously looking at what the cost of those goods are, what the market can afford, and often the market is defined by what external funders can do, like GHIA, Global Fund, and so on. And then in addition, what currently exists or what needs to be improved in terms of the infrastructure to do the diagnosis and treatment and so on. So yeah, we look at the whole suite of things that relate to access and drive towards that result. But it does include, at the end of the day, it does include rather detailed negotiations around price and volume commitments. Yes?
You mentioned several issues of the negotiation of IP rights. So my question is, what are those issues that the parties are most concerned about and most often disagree upon, and how to manage such a negotiation on these disagreements in face of academic agency?
Yeah, so usually, so on the question of IP rights negotiations, the issues that are negotiated usually fall into a couple of categories. One is the geographic scope of where our global access obligations are intended to apply, which would mean that that is the scope of markets where we expect that the price is going to be low. And so, in connection with the IP licensing, that's one of the elements of the discussion. And then, the other is, getting to this point that we made earlier, and that is, the field of use. Is it going to be limited just to, the particular application that we're funding, or more broadly? And, we have discussions about both of those issues, and, in the end of the day, they are, 99.9% of the time successful. Yes?
I had a question similar to the first one that was raised about the decision-making process and how the foundation decides which issues to prioritize. So, naturally, in a disaster or emergency situation, you're going to quickly focus your resources there. But just curious how that decision is made, and when you focus on that immediate emerging crisis, as you alluded to, it detracts from the ongoing humanitarian crisis in a place like Sierra Leone, etc. So, in kind of weighing all the different factors in your mind, I guess, what are some of the stronger influences and more powerful actors when trying to decide which issue to focus on?
Yeah, so that's a good question. I think. Well, let me just say one thing at the outset is that what we experiences I described when we responded to the Ebola crisis, and the fact that we had to allocate, personnel, including myself and a bunch of others, to work on that rather than other things, it did detract from sort of the day-to-day business. And that for us was a learning experience. We hadn't gone through that before, so I wouldn't say it was a surprise, but I think that sort of the scope of the issue was a surprise internally in terms of management. But I don't think that we would have changed the decision to do that. As a matter of fact, we would continue to look at those possibilities. But in our work as a whole, it's based upon a number of factors. Both what we perceive as the area in which we can make the greatest impact. This kind of general statement that doesn't really say that much, but we work very closely with institutions that identify what the global burden of disease is and look at areas where the burden is high but the response has been lacking, and what's missing is new technologies, drugs, vaccines, diagnostics, and say, okay, that's an area that we can make a difference. And so that's why we focus on intermittent diarrheal disease, we focus on malaria, tuberculosis, HIV, and so on, because those are areas generally, and then within them, certain projects that we think are underrepresented. There's other institutions like Guinea worm, for example, is an issue, but the Carter Institute is doing a lot on that. We do support some of that activity, but given that it's being addressed, otherwise it's not something that we invest as much in as we do other areas. And so in that sense, yeah, we do work with others to both identify what we should work on and then make sure that we're coordinated. So work very closely with the World Health Organization in that sense.
I'm going to stop us right now just so we do get some break. Thank you very
Thank
much.
you.
That was good. All right, try to come back in 10 minutes. Try to meet people that you're meeting to meet here and grab a cup of coffee or something and we'll get started again.
No, because Vicky actually and him actually created so it's out of Kenya. I guess it's was that also okay. Yeah. We've been working very closely certainly in Liberia and also they fly in a lot of the seminars and actually that's some of the entrepreneurial pieces and stuff for us. And very I don't know, it just seems like Kenya, the Eastern African side is really it's like, so they've all brought that up. Because I know Duke does a lot of Tanzania stuff. First 6 weeks in Sierra Leone, I was able to work with Dr. Nathan Thielman, who's and just talking to him and just resources. So very much so. I know Duke is working with Sierra Leone now to get funding to try to start some sort of research institute.
That's good. That's good. I also prefer to see one.
Okay. And is that why you were looking to take those other steps Yeah. Well, I would definitely say that, after working in Sierra Leone for the last year and a half, it doesn't come without friction. I think all of Africa is a lot of friction, but I think if you're able to address it, you're able to get just get the data, synthesize all that stuff out so you can actually move forward.
Although, I mean, even before, so hopefully there will be a Yes, yes. So a lot of companies, a lot of the big companies, still United States. Okay, good. Yeah.
You got there? Okay, good. I mean, I was going to send an email out to all of you, just to be great in terms of love is amazing. For example, one of my students yeah, so his personal story. Yeah. I am going to say something.
So you got a lovely guy. Like oh, okay. I'll take a
CBS.
that I was still looking at.
Right?
Great. Good. Busy. But that's good. Yeah. So yeah, it's a very mid-sized company. See you later. We don't but it's the same, essentially the same building. Make a product, we're not going to like it's not working. So, all the products out. And then what I'll talk about is how do you use all of that? Commercially sustainable. That's very different.
And then you all have to do the additional step. You all have to we have asked for the issue there all has some legal restrictions on what they can pay for. As an example, they can pay for touches and products, but not for equipment that doesn't. So, wow. 75% of this actually gap that they legally cannot pay. And this is where that's what we're going out and saying, how can you help us? How can you come together we have a sustainable operation?
Alright, thanks, Julie has one more.
That sounds like a good plan. Let's go around first way, if you don't mind.
So that's so that's going forward, there's salaries, supplies, contracts, all the things that you do to services to keep the facility going, because it's a multi-decade operation. It's not going to so that's its ongoing that cost covers all of the associated costs of doing the development. So that at the end of 2 years, we expect, if we got it going today, 2 years from now, we expect to be able to say, here is a vaccine that is ready for no something about the vaccine, response of the safety profile of the vaccine, and either we stockpile it or we transfer it to somebody who wants to take it. Make it
because it was used for regional passes. 2 to 3 hours. Yes, yes. Well, those costs are built in.
It's not like you worry about. Get a glimpse of Durham. It's funny. One of the I'm just kidding. Well, it's so nice to fly out. Yeah. How do you know him? He's very expensive. Yeah. Super small.
So the clinical research that you mentioned to me that is that related to I am interested. I'm a senior, neuroscience, mental health, I've done a lot of science research, and a competition related to neuroscience. But I'm also interested
Excuse me, we're going to get back to work. Critically increasing more networking on our lunch.
Learning
grab your coffees and. Sorry, presses.
Yeah.
Well, enjoy. Good morning, potentially don't know much. Yeah. I know. Yeah. And I was like, alright, well, I did that. And just like gave me so there's a lot of yeah. I definitely like I just got luck. I was like
Okay, we really are starting back this time. Interviews will have to wait for lunchtime now. So I'm excited about our next speaker. She participated in our workshop in November and has some amazing ideas, both on how Merck is putting together their Ebola vaccine efforts, but also how partnerships and alliances can come together more quickly in the future. So I hope you welcome Dr. Swati Gupta. She's an epidemiologist trained in infectious diseases. She's the executive director of the Office of Public Health and Science at Merck Vaccines. Her Bachelor of Arts degree is in 1995 from Washington University. Her master's in public health is from Yale University School of Medicine, and her doctorate in public health is from Johns Hopkins Bloomberg School of Public Health. At the present time, she's working at Merck Vaccines on the development of innovative partnership models to address cross-cutting issues related to vaccine science and technology. As part of this role, she's working with key external stakeholders to facilitate accelerated Ebola vaccine development efforts to enhance preparedness for the current public health crisis and for potential future outbreaks. From 2000 to 2014, Dr. Gupta was in the Department of Epidemiology at Merck Research Labs. She's worked on many different diseases, infectious diseases. I'm not going to list them all here, but she also worked at the Bureau of Tuberculosis Control at New York City Department of Health, and her research interests include longitudinal cohort and database studies to measure incidence, prevalence, and biomarkers, risk factors for infectious diseases and vaccines. So, Swati, welcome.
Great, well thank you, Julie. It's really exciting to have the opportunity to speak a little bit about Merck's Ebola vaccine program this morning. And I wanted to thank Julie and her whole team for inviting me. And I should mention before I start that this presentation really is on behalf of a very, very large external group as well as a large internal team at Merck. So the title of the talk is Merck's Ebola Vaccine Program, Unprecedented Pace, Partnerships, and Public Health Imperative. So I'm just going to talk a little bit about the status of the epidemic, the ongoing epidemic, because I think that's going to be relevant later on in the presentation when discussing efforts to contain the current outbreak prior to having a licensed vaccine. I'll spend some time discussing the partnerships because I know that's of interest and all of the various partnerships that facilitated and expedited our development. And then I'll just speak briefly on an overview of the vaccine candidate itself and what the development program looks like, where it's at, our plans for product licensure as they stand right now, as well as some of our pre-licensure efforts to contribute to extinguishing the current outbreak, and planning for future outbreaks. And then I'll go into some of those lessons learned that I think everyone is interested in, just touch on a few topics that, areas where I think there could be improvement in the future. So most of you probably are already aware that the first cases of Ebola or more recognized cases, I should say, were in March of 2014 in Guinea. We know that EBD is caused by Zaire Ebola species in this particular outbreak. And cumulatively, there's been over 28,000 cases. I think we're getting close to 29 now, and over 11,000 deaths. The most heavily affected countries include Guinea, Liberia, and Sierra Leone, although there were a few cases in Nigeria, Mali, Senegal, Spain, the US, the UK, and Italy. And many of you probably saw various declarations of the outbreak being over, depending on which country it was being spoken about. So on January 17th of this year, Liberia was declared free of Ebola. On March 17th, Sierra Leone was declared free of Ebola, but many of you might have seen that on that same day, there were new cases that were found in Guinea. So we've been working with the WHO on sort of this latest resurgence, and there are over 800 contacts that are being vaccinated, and they do believe that there's somewhere around 5 confirmed and probable cases, and that number may go up. So the local health authorities there have reactivated their emergency coordination mechanism that was in place originally during the height of the outbreak. This is in a region called Nzera-Kore, and there is sort of kind of a rapid intra-agency response that's been in motion over the last couple of weeks. And so the WHO has redeployed dozens of epidemiologists and surveillance experts, contact tracers, vaccinators, so a bunch of people to kind of help control the effort. So in terms of Merck's involvement in Ebola vaccine development, towards the end of 2014, we entered into an exclusive worldwide licensing agreement where we assumed responsibility to research, develop, manufacture, and distribute an investigational Ebola vaccine candidate, which we now call V920. So you may see that through the slides, so you know what that stands for. And Merck and Newlink and a vast global network of partners have been collaborating in unprecedented ways with a focus of speeding the research and the development and the deployment of a well-tolerated and safe vaccine. And so, we do feel that the efforts of all of our partners in the midst of this outbreak really highlights what we can do together as a public health community and what we can accomplish if we work together. So I'll spend some time sort of talking about the program and what has gone well. I'll certainly then come back to some of the messages at the end in terms of, areas where I think that we could all evolve and improve in the future. So who are some of the partners that are working on this large international effort? We keep using this word unprecedented, but it really is. So there's been many different people who have come together. So before I talk about the clinical program, I thought it would be helpful to mention who are the partners and what each group is contributing to that process. So, as you all are probably aware, typically in our vaccine development efforts, for most programs, we tend to do the clinical development ourselves. We fund that effort, and then we have a good idea in terms of what the demand is going to be for that vaccine after licensure. For Ebola, it's sort of been completely the opposite. It's largely an externalized program. Most of the work is being done outside of the company. It's being funded outside of Merck, although Merck is also funding it as well. And then the demand for the vaccine is unclear. So it's been very unique for us, and what's on this slide is a list of some of the organizations that we've been partnering with. Up at the top are 2 of our major partners. So in terms of the development of the vaccine itself, it was originally developed by the Public Health Agency of Canada. They then licensed the vaccine to a small biotech in Iowa called Newlink Genetics, and then it was licensed to Merck in the fall of 2014. So if you look at all the groups that were involved in the development and how this was sort of pieced together, There are a total of 8 separate phase 1 studies. So these are all separate protocols. Usually in a program, ideally, we would develop one protocol and then that would be done at multiple sites around the world or in a few select geographies. But these are 8 separate studies. And one of them includes, there is a WHO consortium that's being funded by the Wellcome Trust. And they have 4 studies that are in Switzerland, Gabon, Germany, and Kenya. There's also a study in Canada that's at the Canadian Centre for Vaccinology. We have one study with the Walter Army Institute of Research. There's one study at the National Institutes of Health. And then finally, we have what's called a 1B study that New Hope Genetics is doing themselves, and that's 8 sites in the US. And then there's been a number of other partners who have been involved that have provided critical input preclinically and in terms of assay development and validation and in terms of manufacturing. And that includes our partners at the Department of Defense, and more specifically DTRA, so the Defense Threat Reduction Agency. There's JVAP, which is the Joint Vaccine Acquisition Program, UCMRID, which is the US Research Institute for Infectious Diseases, and then of course BARDA, the Biomedical and Advanced Research Development Authority, that's part of Health and Human Services. So you can see, a very large cast of characters that have come together to forward the development. And then in terms of the phase 2-3 efforts, if you sort of look at the orange green box on the right-hand side, in Liberia, you have what's called the Liberia NIH Partnership. In Sierra Leone, that study was put together by the CDC and the Sierra Leone Medical School, and BARDA is participating in that as well. And then in Guinea, there's a number of partners for the ring vaccination trial that Rick mentioned earlier, so that's the WHO, Norwegian Institute of Public Health, MSF, and Health Canada. And I'll talk a little bit more about those studies. And then of course our Department of Defense colleagues also and BARDA contributed greatly to those studies. So you can see it was a very large number of people who came together and a number of people to collaborate to sort of push the development along. And so I'll talk a little bit about that more at the end in terms of now, sort of the wonderful part of that, but also the challenges of that in terms of trying to bring all the data together for some sort of package for licensure filing. So most of you probably know that the typical timeline for vaccine development is quite long. It can be somewhere from 1 to 2 decades, and sometimes and often it's 2 decades or longer. So if you look at the timeline for what's actually happened for this program, it's pretty remarkable. This slide is showing some of the larger milestones for the Ebola vaccine program. You can see if you start at the left-hand side that the first phase 1 trial started in October of 2014, so that was before we were even involved. In November, we licensed the vaccine candidate into Merck. By January of the next year, we had made a dose selection decision for the phase 2-3 studies. So a very rapid progression there. And then by February, March, and April of 2015, the WHO, the CDC, and the NIH started their various studies in the 3 most heavily affected countries. So, in vaccine, terminology, or in the vaccine world, it's a very short period of time to go from first in man to phase 3 efficacy trials. And we felt fortunate that our vaccine was included in all 3 of those trials. And they do have very different designs, some of them with placebo controls, some of them not. So I can speak a little bit about that later. And then at the end of July of last year, we did have the interim analysis results from the WHO ring vaccination trial in Guinea, where they found 100% efficacy. And I'll go through that data a little later in the presentation, but we're obviously encouraged and excited by those results, and we're now thinking about our next steps in terms of licensure of the vaccine in the longer term, which is still going to take some time, but also vaccine access in the shorter or medium term. And then the sort of last green box is in August of last year, Merck started what we call the phase 3 safety and lot consistency study. So that's where you have to show for a live viral vaccine that you have consistency among different lots. And then in December, we filed what's called an emergency use assessment and listing, or an EUAL with the WHO. This is a new procedure that helps to make vaccine available in an emergency situation prior to licensure. So really it was all these groups working in parallel as opposed to one entity like Merck working sequentially that's allowed us to expedite the development program. And then this map shows where all the studies are being done all around the world. And so the phase 1 and 1B studies are sort of in the gray circles, and then the phase 2-3 studies are shown in green. And so again, it's a fairly rapid and comprehensive effort by our external partners, considering that the first phase 1 patient was enrolled in October of 2014. And they really did a phenomenal job in terms of all of the community advocacy that they did on the ground before these trials started, and in the phase 2-3 studies, working in countries that are war-torn and did not have great infrastructure. In Guinea, there was quite a bit of violence before the trial. So what they accomplished is pretty amazing, and I don't think that there's too many people who could have done what some of our partners did in those situations, and I do think there's sort of a lot of lessons learned from that. So this is just a quick slide on the vaccine itself. How is it constructed, and what does that genome look like? So the vaccine is what we call a live attenuated recombinant vesicular stomatitis virus, or RVSV. And the way that it's made is that the VSV glycoprotein, which we call VSVG, is in an attenuated VSV strain, and it's replaced with the Ebola Zaire glycoprotein. So then it actually replicates using the Ebola glycoprotein and expresses it in the course of the replication cycle on its surface. The vaccine uses what's called the Zaire Kikwit strain, and we do believe that from a lot of preclinical data that was done earlier before the outbreak, and with the addition of our clinical efficacy data, we do believe that a single dose of the vaccine is going to be sufficient for inducing rapid and protective immune responses. And we think that's going to be important, especially in environments that are resource constrained in outbreak situations. So, the goal of any clinical development program for vaccine development is to evaluate the safety, the immunogenicity, and the efficacy to support the product licensure. And for this program, our goal was always to support licensure in time to affect the current outbreak, if we could, and then as well for potential future outbreaks. And the typical requirements to get a vaccine licensed include an adequate safety database that has to be quite large, with a lot of detailed data. There's also the need for demonstrating the manufacturing consistency, and I think this is a very large piece that is probably underappreciated but takes quite a bit of time and is very much a bottleneck in vaccine development. And then you have to, of course, demonstrate that it works, that there's clinical benefit. So, for an outbreak situation, there are a couple of different options in terms of your regulatory pathway. So, of course, the first one is the direct demonstration of efficacy or effectiveness during an outbreak. So you actually have the opportunity to get the trial started. There's enough cases in your two groups to show that the vaccine works. There's another pathway that's called accelerated approval, and that's where you basically bridge human immune responses with immune responses that have been demonstrated to be protective in animals. And then that involves a commitment after licensure to show post-effectiveness. So there's sort of two components of that. And then you also have something that's called an animal rule. That's another pathway to get a vaccine licensed. I believe it's for vaccines. It's only been done for anthrax. And that's when you can't establish that immunological bridge, then you have to then show that the vaccine works in 2 different animal models. It's quite a stringent path and not one that we're pursuing. So we are looking into and working on the first 2 pathways on the slide. So this is showing just an overview of the 8 phase 1 clinical studies that I mentioned earlier, and you can see who's conducting those studies, the size, the description of the trials, and also the number of different doses that we studied early on. So, it just gives you a range of sort of the full phase 1 program. And I wanted to mention for the study at the bottom of the slide, the Gabon study that's being sponsored by the WHO, they actually enrolled children and adolescents as well. So that was actually our first pediatric data. So not a very large sample size, but at least allowing us to get some idea of how the vaccine works. effects in a younger population. So this is just showing what we call immunogenicity, or immune responses from the people who participated in the trial and they received the vaccines. And this is sort of a combination of data from our North American sites, so that's the NIH and the Walter Reed site and then the Canadian site. And it uses a particular type of clinical assay that's called an IgG-GP ELISA. So, just looking at the two axes on, the y-axis, what you're looking at is the titer, what's the sort of immune response in that person or in that cohort, and then on the x-axis is time. And then if you look at the different colored bars, those are showing you the doses of the vaccine. So you can see by the red and blue, green, and yellow bars, different doses of vaccine that were administered. And basically what's being shown here is that, using the 2 × 10⁷ dose, which is shown in the yellow, you do have a statistically significantly higher GMT if you look out at day 28 than the other 3 doses. And you don't really have a difference between those other 3. So this was information that became available, I believe, somewhere around January 29th of last year, and we made our dose selection decision on January 30th, and then started the preparation for making sure that we could get the vaccine to the field for those phase 2/3 sites. So it was very lucky for us that we felt like we had data that at least gave us some clarity to move forward. This is also showing some more immunogenicity data at just one particular phase 1 site. This is our NIH site, and here they studied 3 doses of the vaccine, and there was also a boost given in this study. And really what the point of this slide is just to show, if you look out to the right-hand side, that we do have durable responses out to 6 months. And we do have now more recent data just giving 1 dose of the vaccine that shows that the responses are durable out to 6 months. And we expect to have 12-month data soon. So, this type of, what we call durability data is going to be important when we talk about different populations who should receive the vaccine outside of an outbreak situation, and how much in advance they can receive that vaccine and make sure that they're still protected depending on how long it lasts. So this slide is showing the 3 phase 2/3 efficacy studies that I mentioned earlier. And I think as Rick already described, the PREVAIL study is a safety and efficacy study where they compared the Merck and the GSK vaccine to placebo. Originally, they were going to have somewhere around 28,000 participants. Because of the waning epidemic, they were not able to do an efficacy trial, but they did do a phase 2 safety and immunogenicity study that we participated in. And this was the only study out of the 3 that had a traditional placebo group. In terms of the study in Guinea, that's called Ebola Ça Suffit, or Ebola This Is Enough. That was, again, done by a number of partners, including the WHO. They also looked at safety and efficacy in what they called an immediate versus a delayed arm. So basically, when a case of EVD was identified, they then found the contacts and the contacts of the contacts. And those clusters were randomized to either be vaccinated immediately or delayed, which was 21 days later. So definitely a very innovative design. It's a strategy that was used in smallpox vaccination, and they made it into a more a study methodology, a clinical trial methodology. And then there's also a parallel study in Guinea that's being done by MSF. So it's a separate study, but it's a parallel effort in frontline workers, and they enrolled about 1,800 people in that study. And then finally, the CDC is doing what's called STRIVE in Sierra Leone, and they're also looking at safety and efficacy in what they call an immediate versus a delayed arm. It's not a ring vaccination study, but they do have a delay. There's no placebo group. Their study focuses on high-risk healthcare workers, and within that, I think they've vaccinated around 8,000 people, and that has a safety component, a safety sub-study, and an immunogenicity sub-study, and that's ongoing. So they're going to be finishing vaccinations a little later this year in that study. So I thought I would just show a little bit of data from PREVAIL, the study in Liberia, because this recently became available. It was presented by the Liberian investigators at a retrovirus conference recently. And what this is showing, again, is some immunogenicity data looking at antibody responses at 1 week and 1 month after vaccination. And if you sort of just look at the second column, what it shows is that about 94% of people met the definition of what we call a responder. And then, if you kind of go to the right, it's just showing that the antibody responses were significantly different from the placebo group. But we were just excited to see some